What the collaboration really is built around is some technology that Monsanto brings to the table, which is centered on DNA markers. DNA markers can help us develop varieties more efficiently. It's a little bit like a stacked deck of cards going to Vegas. If you can pull out the card every time, you're going to win. And so we're following key genes that are important to us. And what's unique about the Monsanto technology is the ability to sort large number of seed based on the DNA markers that those particular seeds contain. So it helps us be more efficient in identifying plants that have all the characteristics that are important to us for developing new varieties of wheat for Kansas farmers. In a wheat breeding program, we're working with multiple traits, trying to put them all together from different parents. And so we'll make a cross between parents that have complementary traits. And so for this example, let's say that we have 6 different genes that we'd like to be able to put together in a variety. Maybe that's 2 for stripe rust, 1 for leaf rust, 1 for Hessian fly, 1 for barley yellow dwarf, 1 for quality. So in this example, the white beads represent plants that have only 1 or 2 or 3 or maybe even none of the genes that we're interested in. So they may have some of them, but they don't have all of them. And so these are plants that may be interesting, but they're not the ones that are most important to us. The blue beads, on the other hand, represent plants that contain all 6 of the genes that we're interested in. So these would have all 6 traits, and these would be the ones that we were most interested in keeping in the breeding program. In a traditional breeding program, what we're doing is we're making a cross, and then we're looking through the plants, looking at plant type, looking at maturity, looking at a ton of different traits. And so we're selecting those plants based on what we would like to see in that variety in terms of some agronomic characteristics. And so most of the plants that we're selecting out of here, in fact, all of them to this point, are white beads. I don't have any yet that have all 6 of the genes that I'm interested in. And that's because the population that we're working with looks a lot like this. There's only about 1 out of every 64 that are a blue bead. And so when we actually are going out there selecting on these other traits, we're not very likely to pick a plant that has all 6 genes in them. So that's where the numbers game of plant breeding comes in. Where some of the sorting technology with DNA markers comes into play is that we are now selecting in a little bit different population. And now you can see I seem to be picking out a lot of blue beads, and that's because that population looks like this. We've been able to sort all the seed based on the DNA markers and select only the seeds that have all 6 genes. Therefore, we're able to be much more efficient in our selection, and our probability of being able to deliver a variety to Kansas producers that carries all 6 of those genes is much higher here than it is in our traditional breeding efforts here. And that's the value that that marker technology really brings to the K-State breeding program.